Cool. So my name is Inka Pleiss. I am the president of the Marine Society this year. And it's my great honor to kick off our very first speaker night for 2022. Before I get started, I just wanted to say a huge thanks to Gemma and Tamsin, who are responsible for getting this awesome event off the ground. And of course, to all of our speakers for attending and whom you'll hear from tonight. So without further ado, I'll just introduce our first speaker. Our first speaker for tonight has just recently finished her PhD on New Zealand Scampi, working alongside and supervised by Andrew Jeffs. Amy van der Reis, for the research, conducted a preliminary analysis of the diet of Scampi using meta-barcoding. She also looked at New Zealand Scampi population structure through genotype by sequencing and particle modelling. Tonight, Amy will talk to us about the biology of the deep water Scampi and the impacts of fisheries on the species. Take it away whenever you're ready, Amy.
Hi, everybody. I've had some internet issues lately, so what I'm going to do for the duration of my presentation is just turn off my camera, just in case that affects the upload speed. And then I will share my presentation. Alrighty. So thanks for having me to present at your deep sea seminar today. Quite excited to pass on what I've learned during my PhD. So I'm really a molecular ecologist, or I do a lot of genetic work. And so, as mentioned, I did DNA metabarcoding and genotype by sequencing, which might be foreign terms to some of you. And so I'm not going to touch too much on the methods or anything like that, but I'm just going to touch on the results and what it has to do with deep sea fisheries. So I did my PhD at the University of Auckland, and subsequently I've done quite a bit of work with some faces you might be familiar with. So I've worked with Emma Carroll on whales and Kendall on parrotfish. I've also worked with Doc on marine birds. We did a collaboration with Murdoch University, and we did a research trip, and that was also looking at deep sea research. And as mentioned, my supervisor is Andrew Jeffs, and I did research on deep sea Scampi with him. So this is what Scampi look like. This is a female Scampi. You can see that she has eggs, or you call that buried, so it's the buried female. And in general, there is limited knowledge about deep sea animals. If you compare it
sorry to interrupt, Amy. We just the screen hasn't changed for us yet, so we can't see the Scampi.
Ooh, okay. Well, did you see any of the screens change?
Only the very first slide.
Okay. Does it look like I'm back in my PowerPoint now, or is it still just like frozen on the screen?
I think it's still just frozen on the screen, yeah.
Okay. I'm gonna just stop sharing and then retry, I guess. Okay, so can you see the PowerPoint again now?
Yeah, I can see the PowerPoint.
Okay.
Just the first page again.
Okay. And so now I've clicked it into presentation mode. So can you see the full screen now?
No, still not, unfortunately.
Okay. Okay, no, that's fine. Okay, I think what I'll do is I can switch over to a different network, but I'll have to end the presentation. So does Mary maybe want to go so long? And I'll try to sort this out.
Yeah, I'm happy to go now.
Okay, I'll just switch over networks. So sorry about that, guys. Let me just sort this out. Cheers.
Cool. Yeah, so we'll just switch over to you, Mary. I'll just give a real quick introduction so people know who you are, although I'm sure you all know who Mary is. So she's our first speaker now, and she's rather well known at the University of Auckland. So Professor Mary Sewell focuses on reproduction and early development of marine invertebrates, mainly looking into how organisms are able to achieve optimal success through reproduction in different environments. Tonight, Mary will dive deep into deep sea organisms with a focus on the New Zealand context. So yeah, whenever you're ready, Mary.
Yes, and for those who don't know, Inka and I are alumni of the same high school, Howard College, and we first met in a Blake event a few years ago now. Okay, so I'm just going to give you because I wasn't sure how many people in the Marine Science Society were marine scientists and how many people were just people who were interested. So I've kind of made this a bit of a general talk, but please feel free to stop me at any point in time. Just by putting questions in the chat or putting your hand up or whatever we can do that. So we live on a planet that's called Earth. So 70% of the ocean is actually, 70% of the planet is actually ocean. So if you were thinking about this, we should really call the planet Sea, not Earth, which is an interesting perspective on things. But on the Earth, there is a large percentage of the Earth is underwater. So if we have a look here on this graph here, you can see nearly 21% of the Earth's surface is between 0 and 1 kilometre in height. Probably not a good statistic for sea level rise. And then the rest of it is actually much smaller. We have the maximum height, of course, is 8.85 kilometres, which is Mount Everest, and then in the ocean we have a very large percentage of the ocean is really, really deep. So the modal frequency is about between 4 and 5 kilometres deep, though the mean depth of the ocean is about 3.2, and we of course go down to the deepest part of the oceans in the trenches. Now there is a project which is called GEBCO Seabed 2030, which has a plan to map the oceans at high resolution. And this, so far in 2022, we've only mapped 20% of the ocean to high resolution. And so the black is where we still need more information. So we've got good information about the planet in the northern hemisphere, in the Arctic, and in parts of Antarctica, but then there's huge gaps of which we don't have good resolution data. And in contrast, we have a very high resolution images that we could get from Mars and from the moon. So we have really detailed information on planets that are many millions of miles away from us, but we don't actually have the same information available about our own deep oceans. And I'm sure that you've all seen this map, which is a very popular one that NIWA put out in 2016, showing New Zealand and its position on the continent Zealandia. Very clearly see we've got some, large plateau areas in the Campbell Plateau, the Chatham Rise, and the going up towards New Caledonia, and then very obviously we have the Tonga Kermadec Trench, which runs into the northeast of New Zealand. But this map is a bit fudged because it actually only has 250 meters resolution. So even though it appears like it's got a lot of detail in fact it doesn't. And in fact, if we look in a bit more detail about the information that is in this map, in fact we can see it's a very similar situation to what I showed you in the GEBCO map, that we have concentrations that we know quite a bit about, particularly in the coastal environments. This is showing depth information and out along the Chatham Rise. Again, these are all areas that are important for fishing that Amy will probably be talking to us about. And the multi-beam swaths that they do that gives really detailed topography information, we're missing huge parts of New Zealand. So even though we can produce a map like this, the actual underlying resolution of what we know about the deep sea in these environments is actually quite low. Now, in New Zealand, we have a very long coastline, and we have a large EEZ, one of the largest in the world. And this is about 15 times our land area. And most of New Zealand's ocean within the EEZ is actually quite deep. In fact, over half of it is over 2 kilometres in depth, and some of it is obviously much deeper than that, because we have a very deep point in the Kermadec trench, which we'll talk about in a minute. The topography is not always uniform. We do have a large amount of abyssal plain, but we also have seamounts, particularly along this area here, the Louisville seamount around here, and also on the Chatham Rise. We have methane seeps, particularly in the Wairarapa Eddy. We have a huge number of volcanoes, at least 30 or 40 that have been described in the Tonga Kermadec Trench. These are underwater volcanoes I'm talking about, not land ones. We have hydrothermal vents that are associated with the volcanoes along this trench, and we have deep canyons, particularly the one of Kaikoura is quite well known, which is where the sperm whales hang out. Now, what do we have living in this really, really deep environment? Well, as we go deeper, we get less and less species. So this is a Russian graph from the 1960s, but probably is pretty similar to what we have now. So here's the number of species on the y-axis, and as we go down to about 6,000 meters, the numbers, really, really low numbers, in the order of, small numbers of species, way less than 20 or 30 species of abyssal macrobenthuses. Some of this might be sampling bias, because it's actually really difficult to sample at these depths. But at these depths, we get large expanses of mud in the areas that are not covered by features like seamounts and volcanoes and stuff. And there are general trends in the type of organisms we get. We get sponges occur to about 2,500 meters, and then we get sea cucumbers, what I did my PhD on, which are super abundant from about 3,000 meters down, and in fact at the lowest depths are pretty much 80% of the biomass. We have a lot of starfish, a few crustaceans, but generally in the area between 6,000 and 8,500 meters, we start to get really, really low diversity. Now, we have used submarines in New Zealand before. We don't own any submarines, so we have to depend on other countries coming here, bringing their submarines to do that. And the deepest dives have been done with Woods Hole's Alvin, which can go down to 2,500 meters. And this has been used to do research along these volcanoes in the Tonga Kermadec Trench. So these volcanoes are actually huge, and it's hard to sort of imagine how huge they are. So this is Monowai volcano here, and the distance across the crater is 10 kilometers. So in my house, which is in the near Mount Wellington, to the Auckland campus, that's about 10 kilometers as the crow flies. So that's thinking about all of the intermediate area being in this crater. It puts these volcanoes into scale. Most of these volcanoes don't reach the surface, but some of them come quite close. And along their flanks, they have found large numbers of hydrothermal vents, which include lots of life, including worms, but also fish and clams that rely on bacteria for their nutrition. Now, I mentioned that the Kermadec Trench was one of the deepest part of the ocean, and this was recently visited by this guy, Victor Vescovo, who is some kind of venture capitalist billionaire kind of guy, and he was really into mountain climbing, and he'd already ticked off climbing all the top mountains in the world. So he decided that he was going to do a project where he would go to the 5 deepest points in the ocean, because he'd already been to the top. So he bought this submarine, which is called Limiting Factor, which is on a ship called the Pressure Drop. And so he went to the 5 deepest parts of each ocean, so the Arctic, the Atlantic, the Southern, the Indian, and he did a few dives in the Pacific. And then while he was in the Pacific, he also went to some of the other deepest points on Earth. So he went to the bottom of the Mariana Trench, he went to the Philippines Trench, he went to the Tonga Trench, and the one that we're going to talk about, which is the Kermadec Trench, which is the one that is closest to us, which is called Shoal Deep, which is 10,003 meters in depth. And this is the image that he shared. Oh, sorry, no, here it is. So these are the places he aimed to go to. He ended up going to this point here. And this is just to show you his submarine. There he is getting into a submarine. 10,000 meters is, remember, there's an atmosphere change every 10 meters. So it's masses of pressure down there. And so this submarine had to be over-engineered to be able to survive at those extreme depths. He's obviously a bit of an adventurer. And this is the image he shared on Twitter of the bottom of the Shoal Deep, which is the only image that I could find that he's shared so far, which basically just looks like a very rocky outcrop covered with very fine sediment. So perhaps not the most exciting of places to visit. He is a very wealthy guy. He does actually help with science. He has scientists on his expeditions, and the submarine can actually collect samples. So while he's being the one who goes to the deepest part of the ocean and gets the records for that, he does actually collect samples that are used for science. And as he's transiting from one place in the ocean to another, he does do this bathymetry swaths with really high resolution equipment. And this is just an example of a seamount that he has been mapped for the first time, that rises from the ocean up to 2,000 meters. So it's not getting anywhere close to the surface. But it is a pretty impressive topography. And so people like him are contributing to that mapping of the Earth. I've been lucky enough to go to sea in ships, and I've been in 3 submarines. The first one I went to was Pisces 5, which was in Hawaii. This is a fairly shallow water submarine. And this was all done before we had digital cameras, so I don't actually have any of my images of my own, except for what are in my own mind. But one of my most impressive sights on this dive was these big sponges. So these are hexagonal sponges that are in big fields in the volcanic muds that are at the base of the Hawaiian volcano. My second subdive was in Clelia, which was run by an oceanographic institute called the Harbour Branch Oceanographic Institute. And on this dive, we were in the Bahamas. It was only to about 800 meters. And my big memories from that dive are these big fields of crinoids. So this is a stalked crinoid here in its feeding posture. And as the currents changed and as the sub moved, the crown of the crinoid shifts so that it was getting its fan into the feeding current. So I remember that quite vividly. But my deepest subdive was done in Alvin, the Woods Hole one off East Pacific Rise in the Galapagos Islands. And this can go to a depth of 2,500 meters, so not the same as the limiting factor that Victor Vescovo went on, but quite deep enough. there's a 7-foot diameter bell that the pilot and 2 scientists go in, and as you descend to the bottom of the ocean, just by ballast, which weighs you down, it takes about an hour and 3/4 to 2 hours to get to the bottom. And after you get past about 150 meters, it starts to get very dim, and then it's very dark for all the way to the bottom until you get down the bottom and you turn on the lights. And then when you have done your science and you return to the surface, you get the ballast is dropped off and you slowly rise up, and it takes you about 2 hours to get back up again. And of course, my memory of that is very much driven by the fact that we saw, black smokers and hydrothermal vents. But the most amazing thing for me was that this is a black smoker, and here's some shrimp and stuff around it. We were actually collecting rift deer, the tube worm, as part of our collections. But this is not quite what I saw, but it was very similar to this. And right along the ocean spreading ridge, you get this brand new ocean crust that has just formed. And if you're familiar with obsidian and how black that obsidian is, this is what new ocean crust looks like. You have just this completely black, it's not covered with anything. There's no sediment, there's no organisms growing on it. It's just a completely black, clean rock that had just come out of the mantle. And that's a memory that I will have for a long time. Now, I'm going to just finish up here by talking a little bit about our challenges. And our challenges in studying the deep sea and even getting a good map of the deep sea environments on our planet are that it actually requires a lot of expensive equipment, and it can take a very long time to get the information that we need about the organisms. Now, when I was at Harbour Branch in Florida, we were about 70 miles south of Cape Canaveral, which at that time was where they launched the space shuttle. And every time they launched the space shuttle, we could actually look out the window of the lab and see the space shuttle take off. And one time I applied for and got a ticket to go to the causeway, which is near where it takes off. And it's incredibly amazing because, it's loud and you see the shuttle go into the sky. The cost at the time of sending the space shuttle into space was $442 million. And one of the scientists at Harbour Branch mentioned in a general public lecture that he was giving that using the costs of Harbour Branch submarines and ships at the time, because the cost of using the submarine includes the ship that launches and retrieves it and all the technology that goes with that. So for the same price as one space shuttle launch, we could have sent Harbour Branch submarines to the bottom of the ocean 24 hours a day, 365 days a year for 116 years. Now, I don't want to disparage what we might learn from, sending the space shuttle into space, but it seems like in 116 years of exploration using submarines, we probably would get a bit more bang for our buck than we got from one space shuttle launch. And I was reading today on Twitter that there's a new thing that NASA is launching at the moment, and it costs $4 billion per launch. So imagine how much deep sea exploration you could do for that. So what we have in the deep sea is we have very limited information. We can't get very good samples. We rely very much on philanthropists like Victor Vescovo, who allow scientists to get samples from his vanity project, but that's not sustainable in the long term, and particularly because of two things. The first is that fishing does a bit of damage to all of these environments. So these are the seamounts that occur along the Chatham Rise, and this is graveyard seamount here. And this was a study looking at the fished and unfished sides of the the seamount. So on the seamount, you have a very diverse, what we call biogenic habitat, very three-dimensional, lots of sponges and bryozoans and all sorts of other bits and pieces. And this, in contrast, is what the fished area where the deep sea trawlers have gone in. So this is a challenge for deep sea environments because we're actually destroying many things that we haven't really looked at in a great deal of detail. And in the next 10 years, we're going to have a whole new issue, and that is because with the transition to cleaner energy, the demand is for many of these rare earth elements and metals, which are found abundantly around the hydrothermal vents. So the hydrothermal vents are often called black smokers or white smokers, and that's because as the superheated water comes out of the vent, in that water, all of the metals are in solution. And just from basic chemistry, as soon as you hit a hot water hits really cold water, all of the chemicals, the metals in solution can't precipitate. And that's why they're black if they've got a really high iron content, or if they're white if they've got a different other chemical composition, which escapes me at this point in time. So in the area around hydrothermal vents, you get high concentrations of these elements that are of considerable value for many processes, including manganese, etc. And we're going to have incredible pressures on the deep sea part of the ocean because they are designing mining ships, which basically have a pump which is going to suck everything off the bottom of the seabed, pump it up to the surface, extract any of the nodules that are there, or and bring them back down, and then have these plumes of sediment that come behind them. And this is obviously going to impact any of the organisms that live on the benthos. So that's my kind of not very good ending, but just as a important thing to think about is that we have deep sea environments of which we know very little about, which we are about to start mining, and whether that's a good idea or not. So that's the end for me. I'm happy to take any questions or debate that you might want to have about that.
Awesome. Thank you, Mary, for that wonderful talk. I guess I'll just kick it off with the first question. So you mentioned the GIBCO Seabed 2030 project, and I was just wondering on whether they whether that mapping is done by ships or by satellite.
It's done by, because you have to do it by ship. So to get that high resolution things, you have to do that deep swathing, which basically they send a signal down to the seabed and then it reflects back up. And so basically you can only get a little bit wider a track than what you've got the ship traveling on. And so basically you need to have a whole bunch of ships going in the same direction, but not in exactly the same spot. And then they can be overlapped. And the problem is that there are, shipping routes between one place and another in which we get good coverage of those kind of things. And then there's lots of areas of the ocean that aren't really often covered by ships. And so we're always going to have a problem with getting full coverage of the planet.
Mm. And in terms of improvements in satellite imagery, that won't advance in the next couple of years to the point where we could then use satellite imagery?
I don't know, but that would be really a great project if you could do that, but you'd have to see through all of those, thousands of meters of seawater to be able to see what the topography is like. Somebody's much smarter than me would have to come up with something like that. But I mean, we have, even if you look on Google Earth, they have a reasonable, some of the features, but every time they go places, they discover new things. Like when I was in the Kermadex in 2016, we were doing, Anil was doing a whole bunch of seabed mapping. The expedition was to do a whole bunch of different things, and Rochelle Constantine was doing a whole bunch of stuff with the whales. And while she was off doing stuff with the whales, the ship was just cruising backwards and forwards doing these, what they call mowing the lawn. So where they go up a track and then they turn around and come back down the track. you know, like they do on fields sometimes where you get that grid pattern. And we discovered a rock that was only 7 meters below the Kanahoe, which would have been very embarrassing to hit, you know, the New Zealand's premier research vessel hitting an uncharted rock. Yeah, so that was a bit dramatic. The crew was like, whoa, crikey, because it was only not very far below the boat. So yeah, that's the issue, is that there's so much that we don't know, and you actually need to be concentrated in where you go and get. So it needs to be, you know, people need to commit to mapping a certain area, kind of thing.
Mm.
But GEBCO's really behind. It's not going to get it by 2030, that's for sure.
Yeah. And we've got quite a few questions in the chat. I'm not sure if you can see them, Mary, or whether you want to.
Yeah. More difficult to study the deep sea compared to space. I don't know whether they're comparably problematic, because the human body is not really designed to go into high pressure environments. So, you know, we've all seen the movies in space where terrible things happen with space suits, and the same is true of submarines. I remember I was telling my 2A class today that when you go in the submarine, the first thing they do is they lock you in there on the deck. So they put the hatch on and put it up, and they leave you in there for half an hour, which is relatively unpleasant, because the Galapagos is actually tropical, and it gets really hot in there, but they're really trying to stress you out and find out if you're getting claustrophobic, because if you do, then obviously they don't want to send you in the submarine for 8 hours to the bottom where there's no way you can get out. So they actually teach you the bell of the submarine can actually come undone, so it can detach. That big sphere at the front can detach from the rest of the submarine, and they actually tell you how to do it. You know, you push this button and that button, and you pull this blue thing, and so you're like, I don't want to hear, because I really have to pay attention to this, but you're like, no, the pilot's going to be fine, there's nothing going to be wrong with the pilot, he knows what he's doing, I don't really need to listen to this kind of thing. But the reality is that they're both very dangerous environments to do things in. I think the deep sea, because it requires ships, can be quite expensive to do, but you know, it is also something that once you've got all the equipment on hand, you can do a lot of it without much you know, extra expense. It's just you know, cost per day kind of thing. So that's hang on. Which was the most comfortable? Oh, the most comfortable was the Clelia, which actually had like seats, like, you know, they have on buses, you know, those plastic seats that kind of have a back. So that was actually the most comfortable, and it actually had a perspex sphere. So it was very, very comfortable. The Alvin, you had to lie down. I don't know if you saw in the pictures, but there's only a porthole like this, and you had to lie down, and it's not long enough for you, and I'm not even particularly tall, I'm only like 170 centimeters, but I couldn't lie like and look out the porthole. I kind of had to lie with my legs sort of crunched up, and you have this weird crick in your neck to look out, and of course you want to look out, but it was not really comfortable at all. We've been working at the Kermadex with a different philanthropist who has his own little sub that we're hoping to get to use in the future, and that even has nicer seats than the Clelia, because it has these little bucket seats, but they're like padded with armrests and all sorts of other bits and pieces. So we're quite looking forward to being able to get onto that. How often do you get oh yes, well I've only been on a submarine 3 times in my 30 years of research, so it's not something that is so easy to do. I just happened to be in the right place at the right time and had a job that was involved in doing this, because in both cases I was hired to do stuff on reproduction of marine invertebrates, and it was that was part of the project, was to do some deep sea work as well. How did you figure out what you're really passionate about before doing the PhD, or did you always know? Well, I suppose cut me off if you need to do other speakers, but yeah. Okay, so when I was 14, I saw this documentary on television. My mother always used to do the ironing on Monday nights, and there used to be this show called Our World, which John Morton, who was a very famous marine biologist, used to host. And there were a lot of bird documentaries, and I really wasn't into bird documentaries. But one night, my mother and I were watching this documentary about Kanahoe Bay in Hawaii, which is on the over from Waikiki. If you go over the other side towards Coconut Island, where University of Hawaii has a marine lab, there was a bay, and it was really quite heavily polluted with sediment runoff and a lot of nutrients coming in from farms. And they were trying to get the coral to come back in this area, and there was some footage of these super long sea cucumbers that were like 7 feet long. And I was like, wow, what are those? I don't know anything about them. And so I went to the library. Pakuranga Library was the big library close to us at the time, because when I grew up in Howick, it was in the country. And I looked up these books on sea cucumbers, and I realized that we didn't really know anything at all. And then and I sort of forgot about them until I got to university, and we were dissecting them in our zoology class, and all of my friends were talking about these. They're just dog turds. They just look like dog turds. How can you think that these are interesting? And they were teasing me so mercilessly about the fact that I was interested in these things. So it was kind of like I had to defend the sea cucumber, which is how I ending up studying sea cucumbers, because I was defending their honor. And it's pretty easy to become a world expert in something that nobody cares too much about. So that's what kind of happened, and that's how I got there. So I didn't always know. I knew I wanted to be a marine biologist, though I had a teacher at high school that when I was failing 7th form English, so whatever that is, year 13 English, and at that time you had to have 7th form English to go to university. And when I was failing in the midterms, this teacher told me, she said, I think you want to go to university, Mary. And I said, yes, I'm going to be a marine biologist. And she said, I think you might need to reconsider your career objectives. And that kind of was like spurred me on, because I just wanted to prove this teacher was wrong. And I wish I could remember her name. I've completely blanked out her name, because I'd love to be able to go back to her and say, I'm a professor of marine biology now. You nearly put me off my passion project. Okay.
That's amazing. Mary, hi, nice to meet you. Great talk. Can you all hear me?
Yeah.
Okay, good. So actually, I'm a PhD student, and I'm studying ecosystem functions. So, and I was wondering how the sediment is probably affected. Well, it's probably affected by the pressure. So would it be different, like if we're talking about nitrogen carbon cycle, would it be different if we went to study down there to what I do in intertidal? And has any work like that been done so far?
Well, Amy can probably better answer this than me, because she studies animals that live in the sediment. But the deep sea is really characterized by really low nutrient input. It doesn't really have much, because all of the nutrients are coming from above, from the productive zone above, and so everything is sinking down towards it, except at hydrothermal vents, where obviously they produce their own through chemosynthesis. But the deep sea is really depauperate in nutrients, and that's why a lot of people think that there's like sea cucumbers and things that are sediment recyclers can manage to live down there, but there's not a lot of diversity in the really deep part of the deep sea. But the deep sea is becoming increasingly recognized as a really important part of the carbon cycle, because of course all the plankton that dies, and it's skeletons and organic material all fall down to the bottom. A lot of it gets consumed on the way, but eventually gets to the bottom, and of course that's the source of where we get you know, our oil deposits that are causing us, you know, untold problems with climate change at the moment. So there is a lot of research, and Melissa will know more about that deep sea cycling of the nutrients down there. So I think that's all my questions. Yes. Anyone else have anything?
Cool. I think that's a good time to probably move on now, just so we stay within our time constraints. So yeah, Amy, if you're ready, we'll just go back to you. And I suppose I did the introduction earlier, so we'll just let you jump right into it.
Okay, great. So hopefully this works now. So can you now see my full screen?
Yes.
Okay, great. All right, so I'm just going to skip to where I was. So this is the picture that you probably didn't see. So these are the projects that I've worked on. So I've done some work for Doc with marine birds and looking at their diet. My PhD was with Andrew Jeffs, and it focused on deep sea lobsters, which I'm going to speak about today. I did some work for Kendall Clements with parrotfish. I've worked with Emma Carroll with whales. And then I did a research voyage in 2019, and we also looked at some deep sea things there. And that was in collaboration with Murdoch University in Australia. So this is Scampi. This is a female Scampi. You can see that she has eggs, and you would say that she's berried because they look like little berries. And so in the talk today, and actually Mary did a great introduction for my talk, because we really don't know much about the deep sea at all, and we exploit the deep sea, and we really don't understand the consequences that it's actually going to have until we actually try to research about it. And so when you compare it to the coastal and terrestrial organisms that are much easier to access logistically and financially, it's just cheaper as well. We just don't know as much about the deep sea. And so we sort of blindly go and take what we want from it. So Scampi are quite the lucrative lobsters. It's a clawed lobster, so you would have been able to see their claws in the little claws in the picture. They mainly exported from New Zealand. They about 67 New Zealand dollars per kilogram, so that's a lot of money. But the New Zealand Scampi is one of 15 species in the Asia Pacific region, but specifically Metanephrop shallendri is endemic to New Zealand. And so I'm just going to give a little bit about the biology of Scampi. And so what you saw in Mary's talk was that sort of muddy deep sea sediment. And that's what Scampi prefer. So they are burrow-dwelling lobsters. So they actually form burrows in the sediment. And so when they burrow, they would also cause bioturbation. Oh, I can't pronounce that word today. And so that is actually them digging, and then the particles from the sediment go back up into the back from the surface back up. And so it's actually quite good for any filter feeders there, because that sediment could contain nutrients, and it's also good for the microbial productivity. So basically just stirring the sediment back up helps quite a bit. And so a bit about the biology of the Scampi. Like most lobsters, they would have a larvae phase, and during this phase they have a pelagic drifting, and it usually goes with ocean currents because they have poor swimming abilities. So, and they're obviously very, very small. And so what you can see here is some larvae at the different stages, and you can see how they develop. But they have a very, very short pelagic larval phase. And so they're only really floating around nearer to the surface of the ocean for 11 days. And this really differs considerably to their counterpart, the coastal counterpart. So if you can imagine the normal rock lobsters we get or crayfish, they have a pelagic larval duration for 9 months plus. And at some stages, they can actually basically circumnavigate the Earth. Whereas here, the scampi have 11 days, and then they drop down to the bottom. And they at that stage, they're post-larval stage, which you can see over here. So they're still quite transparent. So they're not as well protected as the adults with their exoskeleton. They typically reside 200 to 500 meters below. And that's also where trawling occurs. And the trawling would be bottom trawling. So like Mary showed quite nicely, trawling really messes up the sea floor as a net is weighted and it's just dragged along the bottom, and it's just going to basically devour anything in its path. So even, starfish, you're going to have different types. There's something called a sea feather. You're going to have that as well there, and it's just going to take everything, and it's either going to get trampled by the net or it's just going to get stuck in the net. And so this is an example of bycatch, and it's called ling. And so my PhD was part of a very big scampi project. The main aim of the project was to develop a sustainable and commercially attractive harvesting methods for scampi. So they really wanted to go away from the bottom trawling, which was so destructive. And they also wanted to see, well, could they develop a land-based aquaculture for this deep sea organism? And obviously motivation behind it was also that the scampi are highly profitable. And so Cawthorne Institute took on a large portion of this project, and they actually got really far in the life cycle of scampi. So they were able to get it through its larval phase, which is a really big achievement. And here you can see a photo. It's not very clear, but this is the post-larval phase of the scampi. I mean, it's really, really small. And this was like a really good success story, because from this stage, you've already gone through so many hurdles. And so now you can really look on to going to juvenile and adult. And so my PhD focused on looking at the diet and population structure, and they're quite essential to aquaculture. If you know the diet, obviously you know what they're going to eat, so you can feed them more appropriately. And regarding the population structure, you need to have a good amount of diversity in the DNA when you breed organisms, so you don't have inbreeding, which could affect the reproducibility and how the future generations would be able to survive. And so this is the scampi management area of in the New Zealand exclusive economic zone. So the exclusive can't remember which way it is now, but the EZ, I'm pretty sure it's the economic exclusive zone, but maybe it's the other way around. Is this outline over here. And we did sampling at these squares that are colored in. And SCI stands for scampi, so it's taken the first two letters and the last letter of scampi. And it's got quite a few management areas. And so you can see that there's lines, so it's broken up. But I mean, these are, unfortunately, there is no information as to why these were decided. So it's just like they arbitrarily drew a line and decided that this was going to be a management area. And what else you can see on here is the distribution range of scampi, but this is only the known distribution range, as the data is typically gathered by fisheries. So if they catch them, they're like, oh, we know it's here. So we don't really know the whole distribution, but this gives us a pretty good idea. And so this is what you would see. This is also freely available data on the New Zealand's fishery website. And it tells you all the different species that are commercially caught, and it goes year by year and gives you the data about how much was caught and what the total allowable catch is for that specific area. And so Mary mentioned the Chatham Rise, which is this section over here. And we can see that this section of the Chatham Rise has the highest total allowable commercial catch. So it's 408 tons. And then the second area that has the most catch would be SCR6 over here, which is where the Auckland Islands are. So that little island is the Auckland Islands. And that's 306 tons. And then it's quite substantially drops down for the other areas. And for example, over here, where you go towards the Comidex, well, they say you're not allowed to take any from there. And so this is what you get when you bottom trawl. And this is taken from a Scampi fishing vessel. And you can see that there's a lot of bycatch. They estimate that Scampi, when they fish for Scampi, it typically represents only 20%, well, less than 20% of the catch weight. And so that's really, really little. And then they've also estimated that for approximately 1 kilogram of Scampi caught, you're actually discarding 2.5 kilograms of other organisms, or even, Scampi themselves that either got too damaged or maybe they're too small, things like that. And so it really just shows you how destructive bottom trawling is in regards to all the species that you are in fact bringing up in one trawl. And so if we take some of the information I've just shared, for the 2021 fishery season for Scampi, there was around about 1,000 tons of Scampi caught. And so if you just look at the approximate of the 2.5 kilograms of discard for every kilogram of Scampi, we see that there's about 2,600 tons of discard. And that's quite a lot. And so on the left-hand side over here, I've just, on the right-hand side, I've shown bycatch species that are often included. And if you want, I'll leave this on for a few seconds, but it's a little bit like Where's Wally? And you can actually see the different species in the picture. And if anybody can spot the starfish, well, I'll give you some sort of virtual prize, I guess, but there's a starfish in that picture as well. Yeah, so if you were able to scan the picture, you'll see that this over here is ling. We also potentially have hoki over here. Then there's a sea perch, what looks like a sea perch, so let's say over here, potentially red cod. This looks like it could be a giant stargazer, but this really just shows the diversity that's being captured when you targeting, one species. Although when the fisheries do go out, they usually do have licenses for multiple species, but it really just shows you the variety that you get when you bottom trawl. And so, as I mentioned, I looked at gut content. I did this with genetic methods, and I'm not going to go into any of the detail, but if anybody is interested in that, you can feel free to ask a question afterwards. But we were able to identify a lot from the Scampi gut content, everything from crabs to fish. We even found some diatoms, algae, everything that's, we didn't expect so much diversity, but we really found a lot. And so what we could determine from that is that Scampi are opportunistic benthic generalists. They're likely to scavenge and prey on organisms. And we also saw that around 50% of the fish species that were identified are commercially targeted. And so the fish in the diet is likely to be discards from bycatch or the discards that are thrown overboard when the fish are processed at sea. And so how does this influence trying to determine the diet? Well, it actually makes it really difficult because we can see that fisheries have really influenced the diet. And to try know what the natural diet is almost becomes quite impossible. And the fisheries have actually had a beneficial effect for scampi, and also other organisms that are that live in the benthic habitat, because it's actually improved feeding opportunities. If you think quite a few years ago, we didn't have the technology that allows us to do this type of deep sea fishing we do now. And so with the addition of all these discards, we really providing these organisms with the alternative food source. And by doing this, we impact the food web as well. And so you're actually changing sort of the ecosystem structure. And that's really important, because if you know anything about New Zealand fisheries, they're really trying to move towards ecosystem-based fishery management. And it's definitely not straightforward, especially because we have influenced it. So going to an ecosystem-based management, you almost have to think, how have you influenced it, and sort of how can you change to protect the ecosystem, but at the same time, what have we done that's already influenced it, and if we stop something, how is it going to impact it? So it's really complicated, and it's definitely not straightforward. And then I'm just going to quickly run through the population bit of the deep sea scampi. And in aquaculture, it's really important in regards to breeding to know the different amount of genetic material and how it differentiates between populations, because you don't want to inbreed. And this was recently in the news, and it's a prime example of why genetics is so important. So the Taupo prawn park has had really bad survival rates lately, and it's because they didn't actually maintain their genetic pool when breeding. So they actually lost a lot of their prawns, and it was very poor in the last few years. And so if you've ever been there, they've actually closed down, so you can't fish for the prawns anymore. And they're actually looking at bringing in prawns from Thailand, so they can introduce new genes into the pool and hopefully diversify the prawns a bit better so they can survive a bit better. And so how does that link into scampi and fisheries and everything like that? Well, when you're looking at the genetic structure, you're actually looking at, you have to think about things like gene flow, and in order for the gene flow to impact, you have to think of larvae recruitment, because that's essentially how over multiple generations you're going to get the diversity in the genes. And so, as I have mentioned, when the larvae are at this stage, they actually have very poor swimming abilities. So you would presume or assume that they're going to travel by current. And so obviously in science, you don't want to assume anything, and you want to try test things. And so what we did is we did particle modeling. And so this is where we had collected scampi from these 5 areas. And so we modeled, well, if you could presume that every scampi larvae was a particle, and we released particles, how would they travel in the current, and where would they end up? Because it's got a very short pelagic larval duration of 11 days. And so they're not moving very far. And so this is what we saw. I'll just play that. So you can see that the particles move quite quickly on the eastern side of New Zealand, and they eventually come around from the west to the east. So we know that they are following the major currents. And then if we look with the particles released from the Auckland Islands, you can see that, again, it's following the major currents, and you can actually see how it goes. There's a really strong current that goes out towards the Chatham Rise, and so it follows that. And that would be after multiple generations, but it does give you an idea of the direction of gene flow. And so one thing to notice in that is that the particles from SCI 1, 7, 2, and 3 never moved south. And that's really important to know because it means that you're not getting new at least from the areas that we sampled from, you're not getting new genetic material to the south. And that's really important because we know that SCI 6A, which is over here, is one of the higher areas where commercial fisheries go, and it has the second highest total allowable commercial catch. And particle modeling was, it actually mirrored what we saw in regards to the genetic differentiation. And we saw that individuals had different, they actually could be grouped by their DNA. So you had individuals in the east, you couldn't actually tell them apart, so they formed one big group. You had individuals in the west grouping together and the south. And so what does that really mean? You know, I've explained why it's important for aquaculture, but what does that really have to do with fisheries? Well, if we look at the two bigger fisheries, fishery areas for scampi, the two bigger scampi management areas, we can see that SCI 3, which had the largest total allowable catch, is 408 tons. And so that's a really big area. I mean, if you see the outline, it goes all the way around there. So that's allowed 408 tons to be extracted from there. And then you have SCI 6A, which is probably, I would estimate, about a third of the area, and you're allowed 306 tons. But if we consider the particle modeling and what the genetic data was telling us, we know that this area in the south really seems to rely on self-recruitment. And so what that means is that it's going to be more vulnerable to overexploitation because it's not getting new recruitment as frequently as SCI 3 over here. So there was always individuals coming down and it's increasing the gene pool, whereas here it's not. And so it's much more vulnerable to exploitation, but it's also a really important area to maintain because if you had to cut this off, it means that the genetic material isn't going to go there anymore. And so then you're increasing the gene pool, decreasing the gene pool for that area. And also what was interesting is research suggested that in the area over here by the Auckland Islands, that those individuals actually emerged more frequently, or they had a higher level of emergence. And this actually would increase their catchability. So when trawling takes place, they're not going to be in their burrows. And so you can catch them more easily. And so a lot more research needs to be done. As I mentioned, there were only the 5 areas that we took samples from that we could discuss results from, which means that, well, maybe there are other areas where the gene flow would come from, for example, for the Auckland Islands and that. But we just don't know until we can actually do more research. And so I've got 2 questions here that has to do with fisheries in the deep sea. Because deep sea is so poorly studied, there's really a lot to think about. And so the first question that you don't need to answer or anything, it's just to think about. But if trawling and the associated discard is having a positive impact on the scampi population, and in fact, possibly other fish populations, what needs to be considered if bycatch was no longer discarded at sea? Because New Zealand is a bit unique in the fact that the fisheries are allowed to discard at sea, but some countries you're not allowed to discard at sea. And then the second question is, do you think that SCI 6A should be allocated such a high total allowable catch? And with the knowledge we have now, do you think that the management area boundaries should stay the same? And so those are just 2 questions that, with what we've discussed, that, it's just quite a bit to think about and it makes it quite interesting. And so I have some acknowledgements, which was for my PhD. But I'm quickly actually going to just go on one last slide because I think there's a few more minutes to spare. Part of the part of the project, and this was done by some colleagues in Cawthron, they really wanted to pot for scampi because in the aim of the project, it was to create sustainable methods for commercial catch. And so they trialed a whole lot of pots. And for those of you not familiar with catching crayfish, it's like, well, you can see it in the second picture. It's like this big, well, container, I guess. And they put bait inside, and then well, on the coast, crayfish would crawl inside, but you could really use it to try catch scampi as well. And they do it really successfully in Europe to catch scampi. But anyway, it failed quite horribly. In how many thousands of hours they went about trialing these pots to catch scampi. They only caught 7. But obviously, you would want to do potting because it's much less damage on the benthos because you don't want to trawl anymore. However, what they found was that it was really successful for ling. So they weren't catching scampi, but they were catching ling, which is also a commercial species. And you only get about 1% fish bycatch. And you obviously don't have the impact of potential seabird bycatch or mammal bycatch because you're no longer trawling and you just hauling them up in the pots. And there's also the really big advantage of the seabed having minimal damage. And so that's like a really positive thing that came out of the study anyway, because even though they couldn't pot for scampi, they've converted this specific company that we worked with has actually changed all their trawling for ling to potting. So yeah, so I just thought I'd share that as well. But thank you very much for listening.
Awesome, thank you, Amy. I think I speak for all of us when I say we learned a lot there that we didn't know about. Personally, I didn't even know scampi existed. So yeah, that's really cool. And we'll just open it up for questions now.
I actually had a question, and it was, how do you think climate change would influence larval dispersal? So you said that the SCI 7 had self-recruitment, and I was talking to Simon Thrush and Jen Hillman the other day, and they were telling me that if there is obviously climate change, but if temperatures increase high enough, our EAC, which goes on our east coast of the North Island, will move more south, and so Dunedin will get more of that current. And so do you think that we would be influenced and they would have like less self-recruitment and more larval dispersal?
Yeah, so I actually did look into that quite a bit because climate change is, a lot of the effects are modeled because obviously we're trying to predict the future. And so what they predict would happen is that this Tasman front, so this current coming in over the west, is actually going to not be as strong. So that would affect larval recruitment. So you wouldn't have the scampi obviously going to their different locations as fast, well, settling, I guess, post larvae as far as they had previously. So that's one effect. And then obviously that's going to influence everything else because the Tasman front turns into the East Auckland current coming down. And in regards to Dunedin, there wasn't, the study was actually an Australian study where they modeled the currents. And there's been a bit done in New Zealand, but I'm not too sure the regarding down south what exactly would happen. But also regarding, and Mary might be able to pitch in on this because I think she has much more knowledge about this, but in addition to climate change and everything that's happening, you also get ocean acidification, and that could also impact the larvae as well. And also with the increased temperature, that could also make them their pelagic larval duration shorter. So there's a lot of impacts that it does have, and it could affect the entire larval recruitment process.
How long do you think it would not last, but I guess Mary, you can chip in too, because this is a really delicious 724 lecture on your behalf. But how long do you think it would take for the front to weaken before we start seeing change? And how do you think we'd be able to note that change? Let's say it's in 10 years. How would you know that there is change? Would we just be not seeing Scampi anymore through like trawling, or would it just happen naturally?
Oh, I'd have to double check the study, but I mean, it was in quite a few decades. I think they were predicting in the next, 50 to 100 years. So it's not going to be fast, but you would, so how we model the Scampi is through recorded data. And so we actually have the current data for New Zealand for the last like 20 years. So you would slowly start picking up on the changes that way, but I mean, it's going to be quite minor and it's just going to happen over time. So there's something called like generational norm, where, it's going to change so slowly that, in, 20 or 30 years time, maybe it's not even going to be realized, and it's only going to be the previous generation that's going to be like, well, that wasn't happening. And I mean, obviously that's also why sciences plays such an important role in this. So yeah, Mary, I don't know if you want to add anything.
Well, if you want to know about the currents, you should ask Melissa, seeing that's her area of expertise. But in terms of the biology and the larval things, the Tasman front is a bit of a wandering current. I think I'm right on that. Kind of goes a bit north and a bit south, so it does. So sometimes it actually will go east and hits the Kermadex, because genetic information from there suggests that that's a self-recruiting population there that gets occasional immigrants from Australia. And then sometimes it's it's very changeable. So Melissa can talk about that and the time frame of that. But in terms of what Gemma's talking about, the larvae, because it has such a short larval phase, and it's non-feeding, right, Amy?
Yeah, that's correct.
Yeah, so then it's particularly vulnerable to anything that increases the rate at which it uses the nutrients that it has to use up. So in terms of 724, if it's got, it's going to be less trophic. So if you have higher temperatures, you get higher metabolic rate, you use up your fuel, and then you may not find yourself in an environment in which there's a suitable sediment to go to. It may not be deep enough, you may be too close to shore or other things like that. So it does get kind of very complicated, but I'll leave Melissa to chime in on the currents.
I'm still confused on the biology. How deep can they live? Because can they hop across the Tasman?
The larvae or the actual adults?
Well, I was just wondering, because most of the currents are coming from Australia, and Australia's seen some of the really big changes in temperature. I mean, if you're looking at a place where the ocean currents have changed, the temperatures have changed, it's that upstream part where we've had less changes. And so I was just wondering if that might affect your supply of Scampi into New Zealand.
Well, it might if the Scampi... well, Mesonephrops challengerii is endemic to New Zealand, and so if it does exist, the species maybe between Australia and New Zealand, it could, but we just don't know if they exist. I guess in the ditch.
Yeah, it certainly is a really fast, in the Tasman front's a fast way to get here. And it goes, it is kind of like a fire hose shooting out, and then it hits the ridges and gets kind of fluctuates around Lord Howe Rise and then the Norfolk Ridge, and then will flick through, as Mary was saying, either towards the Kermadex or then sometimes down into the East Auckland Current. So it is quite variable, but then you do have those shallower regions in the subtropics that if they prefer to live there.
Yeah, so the trawling data, at least I got back, is that they typically below 100 meters, and their larval phase is only 11 days, so they're not moving far at all. Yeah, I can let me just, I'll share the screen again quickly, and I can show you if we did the 11 days, they would only really move, I would say, like it's possible that they could move this distance in one generation. So they're not necessarily going to make it, I guess, across the Tasman unless there is actually, you know, like stepping stone populations for them to survive.
Yeah, okay, oh, interesting.
I think we'll go to the question in the chat and then we'll probably have to move on because we're running short on time. So Amy, can you see that question or did you want me to read it out?
No, I can see it. Yeah, so I wouldn't, it depends what you mean by default boundaries, because some of the fisheries actually have different boundaries. So, you know, you always would presume they're going on the most information they could when they're deciding, but I can't find anything about how the boundaries for Scampi were decided. But yeah, so I hope that the information we provided would maybe revise the fishery boundaries, but it's a bit difficult to say because, as I said, you know, we only sampled the 5 populations. So you would need quite a lot more data, but it's really important to inform fishery management the work we have done, so at least they have something compared to, I guess, what was previously known.
Awesome. Thanks so much, Amy. So we'll just move on to our lucky last speaker. Melissa, I'll just do a real quick intro so everyone kind of knows who you are, although I'm sure many of people have already had you at uni. Melissa's research focuses on the physical state of the ocean and how and why it changes over time. Melissa is currently the director of the joint graduate school between NIWA and the University of Auckland. And tonight she will be sharing her research on deep water within Antarctica, as well as her involvement with the Antarctic Science Platform. Whenever you're ready, Melissa.
Great, thanks very much. Thanks very much for inviting me. I'm just trying to share my screen. I think, there we go, managed that. Yes, I'm going to be talking about the deep water, but the deep water where it's leaving the Antarctic. So dense water leaving the Ross Sea. This is work that was really building on some work that a US team did. Arnold Gordon and Bruce Huber at Lamont in the US went down to the Ross Sea and made some measurements in some really critical areas, and we're building on that in this project, and also on the Italian Antarctic program, which has been making measurements in the Ross Sea as well. So I've got some other co-authors there, Pasquale, Pierpaolo, and Giorgio, who are all in Italy. Originally, we wrote a proposal to the Deep South National Science Challenge, and that's what funded the original observations that I'm going to show you. So that was me and Denise Fernandez and Aitana Forsum-Vakis. They're both former PhD students of mine. Denise is now a scientist at NIWA, and Aitana is a scientist at Met Service. So it's possible to get jobs in New Zealand in your field. And then we also have an Australian collaborator looking at the water as it goes further to the west. And as you mentioned, this work's now part of the Antarctic Science Platform, and I'll talk a little bit about the questions that we're looking at in that particular project. It's a little bit of a shift from when we originally proposed the work. Many of you have taken Marine Tour 2 with me, I'm sure, and you will recognize this plot. It's looking at temperature in the Atlantic. And so this ship went down the middle of the Atlantic in this plot, dropping a probe as they went. And this is a contour plot of temperature. So the Antarctic's over on the left, Arctic's over on the right. It's a bit hard to see the color scale and numbers, but purple and blue is cold water. And you can see below about 1,000 meters, the ocean is below 10 degrees. And in fact, if you take the average temperature of the ocean, it's 4 degrees Celsius. So the ocean is actually very cold. What that means is that water was last in contact with the atmosphere, either near the Antarctic or in the far North Atlantic and Arctic. So it's coming from contact with the atmosphere near the poles, and it's filling up 60% of the ocean volume. 40%, so most of that is actually coming from contact with the atmosphere near the Antarctic. So 40% of the world ocean volume is filled with what we call Antarctic bottom water. And there's a picture of the thickness of Antarctic bottom water through the world ocean. So those dark blue colors are 2,000 to 4,000 meters. If you think about the average depth of the ocean being 4,000 meters, that means at least half is filled with this Antarctic bottom water. If we take that volume and divide it by the rate at which we think it's being formed, you come up with a replacement time of about 900 years. So a really long time scale that water would get replaced. That's a very simple way to look at it, but obviously if you're in the North Pacific, it's going to be a lot longer before that water is replaced. We think it's maybe 3,000 years there. And if you're off New Zealand, that Antarctic bottom water is coming by maybe 10 years after formation. So it's quite a range in terms of thinking about that water renewing, depending on where you are. That dense water, I've been talking about temperature, so we're thinking about heat there in the overturning circulation, but it's also carrying salt and gases into the deep ocean. So in terms of salt, we can see that that water is getting fresher with time. There's a picture down here from a paper. They looked at very sparse shipboard observations. Can't tell how sparse the observations are in there, but there's only a few ship tracks for each of those little areas. And you can see the blue colors show the water freshening. It's freshening closer to the Antarctic and particularly downstream of the Ross Sea. This top panel shows change in oxygen. And you can see it's all these red colors. That means that oxygen is getting lower in that deep water. And that's true also near the Antarctic in that Australian Antarctic basin, which you can see just south of Australia. Not too sure about CO2. It looks like it's a small contribution compared to what's happening in the Antarctic intermediate water, so in the Southern Ocean further north from the Antarctic, but it also may be a component of the carbon uptake that's changing most rapidly, given the changes in sea ice and other things. So that water that's coming from all around the Antarctic, about a quarter of it is coming from the Western Ross Sea. So just on the western side, and I've got a picture of the Ross Sea over there in the bottom right. So you can see that there are some big troughs in the Ross Sea. So the shelf part is the light color, and then you can see Drygalski Trough, Joides Trough, and the Glomar Challenger Trough are these deeper indentations on the shelf that go out to the deeper ocean, which is that blue color at the top of that plot. So that dense water that is being formed on the shelf from contact with the atmosphere is coming out primarily of the Drygalski Trough, and it's forming 25% of this Antarctic bottom water. It's being formed in Terra Nova Bay, and what's happening is katabatic winds are coming off of the Antarctic. They're pushing sea ice away from the ice tongue and the shore, and then that exposed water is freezing. When it freezes, it rejects the salt, giving it to the water below. That water is becoming saltier and denser. So Terra Nova Bay is this big pot of dense water. You can see it's quite deep, and then that dense water makes its way out of the Drygalski Trough out to the slope and then into the deep ocean. So that trough is shallower than the bay, so it needs to be at that level to make it out of the trough. That water, we know it's changing just from the observations that have been put together. And this is actually a paper that just came out. Well, it hasn't even been in its final published form, so it was just a few weeks ago. And it's putting together some older observations to get a really long time series of the salinity in the Ross Sea. And you can see the salinity, so the scale of part per thousand or PSU, going down. There's a downward trend over time, starting from the late 50s up to present day. You can also see some variability jumping up and down. And we'd been looking at a much shorter record until recently, and that shorter record, we could see that there had been a trend, and then there's been this big rebound in salinity that happened in 2014. So that's one that people have been looking at in the last couple of years and linking to changes in the amount of sea ice that are coming into this shelf region and therefore changing what's happening in that polynya. But this paper that really puts it in perspective, that we're seeing a freshening on these shelves, and that's presumably due to ice melt, not the Ross ice shelf itself, but ice melt further to the east off of the West Antarctic glaciers. The dense water, when it's coming out, comes out as plumes. So these are little pulses of dense water that flow off of the shallower shelf and out into the deep ocean. I've got a picture there from some measurements taken by ship, and the Ross Sea is over on the left-hand side of those plots. So I'm looking at those plots over on the right. Oh, there's my cursor. Good. Over on the right-hand side. So this is the Ross Sea shelf, shallower area. This is the deep ocean. The top shows temperature, so you can see all those cold temperatures of that dense water as it's coming out, and it's flowing next to the bottom. So it comes out and it flows down the slope for a while, but the Coriolis force turns it to the left, and it ends up flowing along the isobaths and out of the region past Cape Adare. You can also see it in the velocity. It's going really fast. It's going about 1 meter per second, so about as strong as the tides if you were out on the Waitemata. So a very strong flow that's coming out in these pulses. The release of this dense water was attributed to the winds from the mooring study. So they put moorings in, and they could see that there were these pulses, and they could see that there was seasonality in it. So it was attributed to the winds by Arnold Gordon. But the Italians had also been measuring in the Dragowski Trough for a while, and there's a picture there of some of the velocities from their mooring. And you can see it's incredibly tidal, and very strong tides over 1 meter per second in the Dragowski Trough, and that's at times mixing the warmer water from above down into that bottom water, and that was particularly true at solstices when the tides are very strong. So what we wanted to do was to go back to those locations where Arnold Gordon had put moorings, and this is in the main outflow where it's exiting the region at Cape Adare, and repeat those measurements, put moorings at the same place with the same instrumentation, and ask a couple of questions. How has that bottom water exiting the Ross Sea changed, and how is the variability in that outflow linked to the tides and the winds? So during the Deep South experiment, we put moorings in 2018 right here at this shallower mooring, and then as part of the Antarctic Science Platform, we repeated both of those moorings, both at the shallower site and at the slightly deeper site. So at that location, we now have 7 years, 4 years from the earlier measurements that the Americans made, and now 3 years from the New Zealand measurements. The Italians were also making measurements in that Dragowski Trough, and so they did that for about 10 years. So I'll show you some measurements from that mooring that was in the trough where the water is coming out. So let's take a look at what's happening to that bottom water at Cape Adare. And on the left, I'm showing you the measurements from 2007 to 2011. And then there's a jump in time, which I'm showing there with that gray bar. And over on the right are the measurements starting in 2018 that went for 3 years. And we've got moorings in there now, so this time series will continue. If we look at the top plot, you can see temperature. And temperature has got 2 cold pulses a year. So the years are these tick marks, and I've put an A and a B next to those cold pulses. And you can see that second one at B is fairly indistinct some years. But when you get to our measurements, you can really see that there's 2 pulses a year of that cold water, which shows that it's coming out of the Ross Sea. The second plot shows salinity. You can see there's 1 pulse a year there, and that's marked there with A. And that's happening in March. And what we think is happening is that dense water is coming down the Dragowski Trough, and then it's being released from the trough in March. The next plots I'm showing are density. So putting temperature and salinity together, we can calculate the density of the water, and you can see that there's 2 peaks in density, most distinct during our experiment since 2018. And then the bottom panel shows the speed of the flow. So this is 50 meters off the bottom, so right in the core of that dense flow. And you can see that it's also got those same peaks. When I got the data back, it was one of those aha science moments. So this data over here is what Arnold Gordon was looking at and had thought that the winds were controlling this dense water's appearance. And what I realized when I looked at it and thought about what the Italians had seen in their mooring is that this release of dense water is happening during equinoxes when the tides are weak. So those vertical dashed lines that you can see on the plot are the equinoxes. There's 2 of those a year when the sun is passing the equator in March and September. And those are when the tides are weak, the tides from the sun, and that's when we get the release of this dense water. We can also put that together with what's happening in the Dragowski Trough, looking at the measurements there. So this is a mooring that's sitting near the bottom, and it's measuring, unfortunately they didn't have salinity, but they do have temperature and flow. And we can see a very tidally driven system. The tides are quite strong, and they've got a very strong equinox solstice pattern. So the gray line that's down in that lower plot shows the tides, and you can see those vertical lines are the equinoxes, and you can see how it's minimum during the equinoxes, and then it peaks in between during solstices. So a really strong variation in the tides and strong tides as well. If we take the velocities and we average over the tides, we can see the subtidal flow. So what's happening if you take out the tides can tell you about the sort of persistent flow at that location. And that's the black line in the top plot. So what you can see there is that when you're at equinoxes, that you tend to have a maximum. So the flow tends to peak when you've got weaker tidal flows. The red line shows you what's happening to the temperature, and that when you've got weak tidal flows, you tend to have very cold temperatures. And then, as the Italians had shown in their paper, when you've got strong tidal flows, you have warmer temperatures because the circumpolar deep water that's coming from the ocean is being mixed down all the way to the bottom. So what this is showing is that the tides are really controlling what's happening with both the flow and the density in the trough. When you've got strong tides, you've got mixing and you have shut down that outflow. And when you've got weak tides, you have more undiluted bottom water and it's able to flow out. If you put what's happening at Cape Adare, which remember that's out in the deeper ocean in the exit region, if you compare that to what's happening in the trough, and that blue line there is what's happening at Cape Adare, you can see that the bottom flows are very highly correlated. And that gave us some confidence that what was happening in the trough, or the water coming out of the trough, was what was showing up at Cape Adare. So what we think is happening is that when you've got dense water, very dense water in Terranova Bay and very weak tides, you have the strongest outflow from the Dragowski Trough. So strong outflow at the bottom and a correspondingly strong inflow above it. When you've got less dense water in Terranova Bay and stronger tides, you have the outflow at its weakest, so a weaker flow out near the bottom, and also the density, the tides are mixing the water, so you have less dense water as well. So that density change between Terranova Bay and the ocean is the accelerator that's accelerating the flow, and the tides are really acting as a brake. The interesting thing is that this means that the inflow of warmer water, which is bringing heat and nutrients into the Ross Sea, should also be modulated. And we actually have some moorings across the Dragowski Trough now to measure that. And that's actually the question in the Antarctic Science Platform, is the exchange of heat and other things across the shelf. Some interesting things about tides. The tides in the Ross Sea are a bit different than the tides that we're used to. We're used to semi-diurnal tides around New Zealand, but the tides are very diurnal in the Ross Sea. And that's why that equinox is showing up so strongly, because it's all about the angle that the sun and the moon are making with the equator. So a bit different than the tides that we talk about in Marine 202. But there are some interesting things about that, one of which is that the moon has a cycle in how far it goes from the equator, and that's the Lunar Standstill, and it's one of the longest cycles we have in tides, 18.6 years, and that shows up in the Italians' measurements in the Dragowski Trough. It's something that's maybe more of a curiosity for most places in the world, but because the diurnal tides are so important here, I think it's actually causing some interannual variability on that kind of 20-year timescale. And indeed, there's a 40% reduction in the tidal energy as you go from minor lunar standstill to a major lunar standstill. So this all sounds very exotic, but actually when you Google it, you find that prehistoric humans knew all about this, and they've built all sorts of stone structures all around the world that show this. It was quite important to them. So I think we should all appreciate this prehistoric knowledge. When you have a major lunar standstill, the moon is going furthest at its furthest angle from the equator. And then we have the strongest tides, and that shuts down the outflow and makes it less dense. So I'm plotting density here. And then the minor lunar standstill, it's doing the smallest angle from the equator, and you end up with the weakest tides and the most dense water and the most outflow. So we're checking this now. This is a model that I made that takes into account that density gradient and the tides. So using the observations to drive a model to come up with a prediction of the outflow. And right now, working with Kathy in Australia, we're looking at the changes in water properties that have been measured downstream in the Australian Antarctic Basin, where a lot of this water ends up, to see how well it can predict some of the big variations that they see. And she's been looking at oxygen actually in the Australian Arctic Basin, and seeing that it varies with the amount of this water coming in. So I think that's all I've got. We see the salinity in the bottom water increasing between 2011 and 2018. So we saw that jump that was seen in the Ross Sea itself. It's been decreasing since then. We see that this release is happening during equinoxes when the tides are weak. And we've got this very simple model to explain that, where the density in Kanahoe Bay is accelerating that outflow, and the tides are acting as a brake. So they're really modulating how that water is entering the deep ocean. And there looks like there could be an influence of this really long tidal cycle on that outflow. So many thanks to the NIWA mooring team who've pulled off these observations in really tough conditions. If I could just say a quick word about the Joint Graduate School. As was mentioned in the introduction, we've got this joint graduate school between NIWA and the University of Auckland in coastal and marine science, as well as having postgraduate projects and supervisors, because we've got NIWA scientists who are closely affiliated with the university. We also have events that we run through the year. At the moment, we've got a seminar series via Zoom. We're looking at careers in marine science. We're bringing in people from different jobs around New Zealand who are out in the workforce after getting degrees in marine science, to talk about their experiences and what they learned and what they do differently and things like that. But we do have other activities that are being planned. At the moment, we've got a student email list, so email me if you'd like to join that. Ultimately, I'm trying to set up a Canvas page. We're in like week 4 of trying to set up a Canvas page with university system, but that's the idea in the end. So you'd be able to look at different events, marine events across the different departments, and get information in that way as well. Great, thanks.
Awesome, thanks for that, Melissa. Super fascinating stuff, and yeah, thanks for actually sharing that. That sounds really awesome, and I'll be sure to tune into some of those events. So we'll just quickly go on to some questions. We've got about 10-15 minutes before we wanted to kind of have this wrapped up. So yeah, I guess I'll just pop in with my first question. So seeing that decrease in salinity recently, has there kind of been a response in ocean currents to that decrease? Have we kind of seen any differences in how that might further decrease with more sea ice melting?
Yeah, we're not sure. We don't have much information about the ocean currents. One of the things that was, we certainly see the outflow changing with the density. So it's decreasing with the salinity decreasing. So we know that the amount of water going in is decreasing with that decrease in salinity. And in fact, there's been some, I think the paper that just came out, they were wondering what might happen if that deep water shuts down entirely, because at some point it won't be dense enough to be bottom water anymore. I've also been looking at that. Because I've got that simple model, I was trying to figure out when it would flip the other way and water would just come into the Ross Sea instead of going out. And I get about 100 years. So using that freshening that has been seen, but you know, that's a lot of things can happen. If there were some pretty catastrophic freshening, then it would happen sooner than that trend.
And is there kind of a way to access a more historic salinity record? You know, like sometimes they can, obviously they have like historic records for sea level. Is there kind of a similar thing for salinity?
I think salinity is quite difficult. The ice cores are probably the best method for going back, a few hundred years, and some of the best things. I guess there is a salinity measurement from those out really small changes in salinity here. So it would be really hard, I think, to do that by many proxies. But you can definitely look at other things that can give you some clues. Like the big thing from the ice cores is to look at, is to infer the sea ice coverage. So that's definitely gone through some big changes, particularly with the change in the westerlies due to the ozone depletion since the 50s.
All right. Cool. Anyone else want to jump in with some questions? Feel free to pop them in the chat. We'll keep an eye on it. Otherwise, I was really curious to know too if you've managed to get out to Antarctica for some of the field work, or whether you've just been working remotely.
No, so there's a lot of people on the voyages. There's a lot of different projects that are planned, and I've volunteered, and I've been kicked off because they have plenty of other projects. I'm really glad that they're still able to do the work. Aitana did get to go on the first one, and because we had some CCD work, some hydrography that we had planned, and needed to make some decisions, so she was able to go on that one. But no, I haven't.
We've just got a question in the chat too. So do you know what sorts of projects are going on in the joint graduate school at the moment?
At the moment, we have these affiliated NIWA academics, and they are offering projects in their areas. So go ahead and take a look at their projects. As I say, we're working on other initiatives and things that you can get involved with that aren't to do with a degree program. But in terms of the degree program, it would be the research of the different faculty members. I hope that answered your question.
Yeah, so Tamsin, most of the marine people at the university are all part of the joint graduate school as well. So I am, Rochelle, and any marine person is sort of really part of that, and our students become part of the programs that that Melissa organises, and my PhD students really have enjoyed some of the work, especially in the COVID times when we were able to travel. They had some really great experiences at NIWA and Wellington that they both really enjoyed. So yeah, hopefully we'll be able to get back and do that.
That's right. We're planning at the moment doing some in-person things and going back to NIWA Wellington to have a visit and run our spring school. Fingers crossed. Okay, Facebook. Okay, I don't use Facebook, I have to confess. I can certainly, it's great to have your email address. I had made contact previously, but I will certainly send opportunities through to you guys so you can disseminate it through the Marine Science Society.
That would be awesome.
Great, I see another question there actually from Leonardo. Are there any opportunities for students to go on Antarctic research expeditions? Usually not undergrads, because the berths are, at least on the ship, so it's usually postgrads that go. I think on land occasionally there are some students that go, but usually on the ships the room is so tight there's only a few postgrad students go.
Leo, if you keep an eye out on the Blake and Vesta ships too, I know one of those goes to Antarctica. So if you're interested in that, that would be a really cool opportunity.
Yeah, the problem is that at the moment with COVID, with them trying to keep COVID out of Antarctica, they've actually restricted the number of science events that can go. So there's kind of a bit of a backlog in terms of science events, and they have really restricted the number of people that you can take on a science event. So that has restricted that opportunity. And also, I don't know if you realise, but they're about to rebuild Scott Base, which means they actually have to dismantle the old base and move it and put the new base. So they're actually in a very low Antarctic event space at the moment, because I'm part of the Antarctic Science Platform too, but I do stuff on the zooplankton as part of that. And yeah, we've had a lot of our science delayed by inability to get to Antarctica, and some of our programs are really behind schedule because we haven't been able to get to do the field work that we need to do. So yeah, it's kind of like not a good time if you want to go to Antarctica at the moment, because even members of my Antarctic Science Platform team, there's no room for us to go either. So we just get the samples when they come back. So.
Yes, so Leonardo, to follow up on your question there about postgraduate students, usually it's students whose project is affiliated with that field work. So they'll be the ones who have priority to go on any of those voyages. We're still talking to NIWA about student opportunities for other voyages though, so non-Antarctic ones. So if we can get that organized, that all works out. It just depends on the availability of berths for upcoming voyages. We'll let people know.
Awesome. Mary, I just want to go back in terms of keeping COVID out of Antarctica. Is that more of a, you don't want people to get it there in case they do have bad reactions because it is so remote, or more because you don't want to introduce it to, say, like the organisms in that environment?
It's about keeping people safe, because even the biggest bases like McMurdo, they have a hospital quote unquote, but it's really just it's kind of like, you guys are too young to see MASH, but it's kind of like MASH, except instead of being a tent, it's kind of like in a building, but it's really rudimentary hospital, and they only had like, I think, 4 beds. So if COVID got into the Antarctic, it would be really bad, because it would spread really easily, because people obviously are indoors most of the time, and feeding in large groups in the cafeteria and stuff like that. So the decision was made by most countries that they would restrict access to Antarctica, and people who are doing research in Antarctica had to do, that were travelling through Christchurch, had to do a 2-week MIQ when we had MIQ, and then they went to Christchurch, and then they did another 10 days isolation in Christchurch after MIQ before they were allowed to go to Antarctica. And then on the way back, they had to do another isolation before they left Christchurch. So it was a really big commitment of time for people to essentially be spending 3 or 4 weeks in isolation in a hotel in Christchurch before you could go to Antarctica. So that was one of the downsides of that, is that they had less flights, there was less room for scientists, and they had a pretty arduous protocol to keep it out.
Amy, did you have a point?
Yeah, it's probably a bit off topic, but I was wondering if New Zealand has any deep sea brine lakes.
I don't think so. So you're thinking like I
posted a link for anybody that wants to have a look, but basically I think they found some in the Gulf of Mexico.
Yeah, the famous ones are in the Gulf of Mexico, but they're like hypersaline, so they're super, super salty. And so I would think that we have a very robust oceanography that we would probably need to have an area that was sort of not in the flow of a great current to have anything. I certainly haven't heard that we do. We do have cold seeps along the Hikurangi margins, and we do have hydrothermal vents in the Bay of Plenty and on the volcanoes that we said, but I don't think we have any saline lakes.
No, very well connected, and there's lots of big ocean currents around. I think the Red Sea might be another place where there's some very salty, deep pockets.
Awesome. Well, if no one else has any questions, I think we will close it there. Thank you so much to all of you speakers. It was, just amazing hearing all of you speak. I learned something new from every single topic, which was really cool. And I'm sure everyone else did here too. I'm sure that for anyone here who might think of questions later, that any of our speakers might be happy to get an email, as long as you're maybe patient with responses, of course. I think we'll close it there. So thanks everyone for attending. Thanks again to our speakers. Thank you, Gemma. Thank you, Tamsin. And have a wonderful night.
Great. Thanks very much.
Thanks.